In this video, we will discuss about the effect of this ARHGEF1 on the immune cells, that is CD8 plus T cells, whether it will cause this increased antiviral and anti-tumor response or this increased cytokine polyfunctionality or not. So we will start with this panel A, where they will check the cytokines, that is interleukin 2 and this IFN gamma, and what they are able to see in the case of controlled wild type, where ARHGEF1 is not knocked down. They are able to see signals of around 3.83 and 3.83 for both this TNF positive and interleukin 2 positive, and around 4.05 for this IFN gamma positive and interleukin 2 positive. And correspondingly, in the ARHGEF1 knockout, they are able to see increased in the amount of this TNF alpha and interleukin 2 positive cells, that is from 3.83 to 21.2, and this in IFN gamma and interleukin 2 from 4.05 to 8.60. So this indicates that there is an increase in the cytokine polyfunctionality with the loss of ARHGEF1. And these things they have also checked in this CD8 plus T cells, that the change in the CD8 plus T cells is seems to be high in the case with metastasis compared to the case with metastasis in this ARHGEF1 knockdown compared to the case with metastasis in control wild type. That is shown here in the points in the numbers, that is 0, 1, 2, 3. That is the amount of 1 is increased in the case of ARHGEF1 knockout compared to the case of control wild type. And when they check the effect of different cytokines on CD8 plus T cells, in most of the cases they see increase in the CD8 plus T cells in ARHGEF1 knockdown. That is this one, first column is for all the cytokines, TNF, interleukin 2, IFN, GABA are used or injected. They see an increase in the CD8 plus T cells, and with this TNF and interleukin 2 also it is increased, but when they use only TNF they see a decrease, but that decrease is not statistically significant. And when they do not use TNF alpha, they do not see a increased and in both the cases. And but when they use only TNF alpha, they see an increase that is statistically significant, and the increase is also there when they use only interleukin 2. But and the only case where the CD8 plus T cells goes down, which is statistically significant, is when they use only IFN gamma. And thereafter they sorted out, use the UMAP to find out different populations that are presented within this ARHGEF1 knockdown in terms of CD8 plus T cells, and they distinguish them in this different CD8 plus T cells populations, that is where the CD44 T plus cells populations are predominantly found, and PD1 populations where they are predominantly found. And in terms of the means, they plotted the heat map where we can see that which populations contains what kind of markers in high amount or in low amount. And when they check the frequency of cells in clusters compared to the wild type, they are able to see that here the example shown here for 4 clusters, that is cluster 4, 9, 10, 12. And what they are able to see is that in the case of cluster 4, the amount of control wild type is high compared to the case ARHGEF1 knockdown. And but for this case of cluster 9, they do not see any significant changes. But for cluster 10 and cluster 14 also, they again see significant changes in the tumors, in which is higher in the case of control wild type, whereas it is lower in the case where ARHGEF1 is knocked down. Thereafter, they wanted to check whether what happens to the exhaustion markers that are usually expressed to suppress the CD8 plus T cells. And one of the exhaustion markers is PD1. And when they check the PD1 in the control wild type, they see expression of around 8.94%, but when they check these exhaustion markers in ARHGEF1 knockdown, they see a decrease in those populations. So here they check this PD1 expressions and this TIM3 expressions. These two expressions they have checked. So both of these expressions goes down when the ARHGF1 knockdown is there, indicating that the exhaustion markers goes down. At the same time, the cytokine polyfunctionality increase, which makes it a good candidate which makes it a good strategy to target the cancer cells. And when they check this PD1 in the case where there is no marker, where there is no tumor, they do not see any PD1 signals. But in the case of tumors, they are able to see a significantly high amount of signals in the case of this this wild type, and compared to the case where ARHGF1 GEF1 is knocked down. And here they check the PD1 and TIM3 plus CD8 plus positive cells. And when they check polycytokines markers, and in the polycytokines markers, that is H2KB over cancer cells, and in this case they check the CD8 plus T cells, and they are able to see that polycytokines markers are overexpressed in the case of this ARHGF1 knockdown, but the increase is not statistically significant. And when they check this tetramer CD8 plus gated cells for the cell exhaustion markers, they can see a significant decrease, almost up to 50%, in the case where ARHGF1 is knocked down. So this is similar to what they have checked in the panel H, but here in the panel J they checked it for this CD8 plus gated populations, tetramer CD8 plus gated populations. And the PD1 and this PD1 TOX positive cells, and also shows that in the case of this knockdown, this is ARHGF1 knockdown, the amount of PD1 plus cells is statistically or significantly lower, and also this PD1 and TOX positive PD1 and TOX positive cells is also significantly lower. So all this indicates that this loss of ARHGF1 will not only enhance cytokine polyfunctionality, but it also suppresses cell exhaustion markers such as PD1 plus and TIM3 plus cells, PD1 plus and TIM3 plus positive markers for the cell exhaustion are results are shown here. So I hope you understood the results that are presented in this figure and convinced by how these researchers have validated their hypothesis. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.